Life has energy. Energy has traditionally been defined as the capacity to do work, that is, the capacity to set an object in motion. There are many forms of energy. Kinetic energy, or the energy of motion. Heat, which is the motion of molecules. Electrical energy, which is the motion of electrons. And radiant energy, such as light. These different forms of energy are readily converted from one form to another. Energy may also be converted to an inactive state, known as potential energy. We can demonstrate potential energy with a steel spring. If we give energy to a spring by pushing it, it will spring back. However, if we keep the spring under compression, the internal tensions are maintained and can be released later. In other words, a spring under tension has the potential for doing work. That part of the potential energy of a system that has the potential for being converted to work is called the free energy of the system. As potential energy is converted to other forms of energy, it tends to become dispersed, resulting in increasing disorder, and is no longer available to do work. The measurement of the energy that is not available to do work because it is randomized throughout the system is known as entropy. A disordered system has little or no potential for doing work, and therefore is low in free energy and high in entropy. In most spontaneous energy-releasing systems, disorder will increase. Thus, systems tend toward a state of low free energy and high entropy. However, it is possible for a flow of energy through a system to maintain it in an ordered state of low entropy and high free energy. In such cases, the entropy of the surroundings must increase. A living system which is highly organized is a high free energy and a low entropy system. A constant source of energy is required to maintain this system. If the energy source is eliminated, the entropy of the system begins to increase. This loss of order constitutes death. The original source of energy that maintains life is radiant energy from the sun. In the sun, energy is made from matter. 4 hydrogen atoms fuse to form helium, and some of the original matter is converted to energy. Green plants absorb radiant energy from the sun and convert it to chemical energy, which is a form of potential energy. The potential energy of molecules is a measure of their relative stabilities, which are primarily determined by the strengths of the bonds that hold them together. Atoms form bonds because of the electrostatic forces within the atom. The nucleus of the atom carries positive charges, and the electrons which move about the nucleus carry negative charges. The attractive force between these opposite charges holds an atom together. A bond between 2 atoms is formed when a pair of electrons is shared by 2 nuclei. If the attractive force is very strong, then there is a strong bond. It takes considerable energy to break such a bond. However, molecules also contain repulsive forces due to the fact that like charges repel each other. If the repulsive forces within a molecule are strong, or if the electron pair is shared unequally, then the bond is weak. A weakly bound molecule will tend to vibrate with a greater displacement, It takes less energy to break its bonds. When weak bonds are broken, more stable molecules are formed. In a living system, a common energy-releasing reaction is the oxidation of a carbohydrate, such as the sugar glucose, to form carbon dioxide and water. Glucose contains 6 carbon atoms in a highly structured molecule. Because of its size and structure, it's a sluggish molecule. When glucose is broken up during a reaction, the products, which are smaller molecules, have higher velocities. Energy is released as heat. Entropy is increased as free energy decreases. This reaction occurs because oxygen has a strong attraction for electrons. This type of reaction, which involves the transfer of electrons, is called an oxidation-reduction reaction. The glucose was oxidized because electrons were removed from it. The oxygen which accepted the electrons was reduced to form water. The burning of wood is an oxidation reaction that releases energy. Glucose and other components of wood are being oxidized rapidly. The system is losing order and free energy. The reverse reaction, the reduction of carbon dioxide to form glucose, requires energy because of the highly ordered structure of the glucose molecule. It requires energy to create order out of disorder. The synthesis of glucose, or other organic molecules, serves to store free energy that can be released later by oxidation of the molecule. A living system obtains energy by oxidation of glucose and other organic molecules. But life is a system of high order and high free energy. The reactions have to be controlled so that the free energy of molecules can be conserved to do work or to drive other chemical reactions. Control in a living system is provided by enzymes. Enzymes are proteins that function as catalysts. That is, they speed up chemical reactions. An enzyme has a specific binding site to which specific molecules are temporarily bound in such a way as to favor a specific reaction. Enzymes may speed up reactions by as much as 1,000,000,000 times. Because of their specificity, enzymes organize reactions into sequences, and in some cases they are arranged so that the reactants can move step by step from enzyme to enzyme. A living system requires the coupling of energy-releasing processes with energy-requiring processes. There are many systems of energy transfer that are possible. However, it would be more advantageous for the evolution of life if living systems had a common mechanism for the transfer of energy, a common currency of exchange. Such a system has evolved, and it's present in all living things, from complex multi-celled organisms to the simplest cell, such as a bacterium. Energy transfer in the cell is accomplished by a particular molecule known as adenosine triphosphate, or ATP. The adenosine end is composed of adenine, a double ring molecule containing carbon and nitrogen atoms, and the sugar, ribose. The adenosine end is the main part that is recognized by the binding site of the enzymes that control the reactions of this molecule. The triphosphate end is the part that contains potential energy. The bonds connecting the three phosphates are weak, partly because of repulsion between the negatively charged oxygen atoms, and partly because the electrons in the phosphorus-oxygen bonds are not shared equally, but are much nearer the oxygen. When one of these bonds is broken and a phosphate is transferred to another molecule, such as water, a stronger bond is formed. In biological systems, the transfer of a phosphate enables the free energy of ATP to be utilized to do work or to drive other chemical reactions. The molecule formed when a phosphate is lost by ATP is called adenosine diphosphate, or ADP. To synthesize ATP from ADP and a phosphate requires an input of energy, partly because of the electrostatic repulsion between the phosphates. In this way, energy can be stored in the ATP molecule until it can be utilized later. Molecules similar to ATP will be split by water spontaneously and release a lot of energy. However, the ATP molecule is protected from being split spontaneously in water by the negative charge on the oxygen atoms. The water molecules sometimes dissociate into charged ions, a negatively charged hydroxyl ion and a positive hydronium ion. The hydroxyl ion is the part of the water molecule that would normally split a molecule like ATP. However, the negatively charged phosphates will repel the negatively charged hydroxyl ion. However, when ATP is bound to an enzyme, the negatively charged oxygens are held down and neutralized by positive charges on the surface of the enzyme, and the last phosphate can be transferred. The fact that ATP is stable in water and needs an enzyme to split it increases its usefulness. The potential energy of ATP is released only where it can be harnessed by an enzyme and is not wasted in a non-specific reaction with water or another molecule. In all living cells, ATP is the common currency of energy transfer. ATP is generated from ADP and phosphate by utilizing energy from the sun captured by the chlorophyll molecule and by utilizing energy from the oxidation of organic molecules. The ATP molecule will then have its last phosphate transferred by an enzyme so that its energy becomes available to synthesize molecules and to power the activities of the cell. Another category of molecules chemically related to ATP assist the energy transfer reactions of ATP. These molecules serve as electron carriers. They aid the major oxidation reduction reactions within all cells. Part of the molecule is adenosine, as in ATP. This part is recognized by enzymes. The electron-carrying part is a nitrogen-containing molecule known as nicotinamide, which is one of the vitamins. When nicotinamide accepts a pair of electrons, they are accompanied by a hydrogen nucleus, or proton. There are two varieties of these molecules that are used as electron carriers. One is nicotinamide adenine dinucleotide, or NAD. The other similar molecule has an extra phosphate and is known as nicotinamide adenine dinucleotide phosphate, or NADP. The process of electron transfer is the primary means by which energy is transferred in a living system, and the process by which ATP is generated. The original source of all the energy that maintains life is the sun, and the process of capturing that energy is known as photosynthesis. The first phase of photosynthesis is called the photochemical phase. When a photon, which is a particle of light energy, strikes the chlorophyll molecule and is absorbed, an electron jumps to an excited state. The excited chlorophyll will disperse its energy by ejecting an energized electron. The energized electron is transferred through a series of carriers, and its energy is used to generate an ATP. The electron, after losing its energy, is then returned to the oxidized chlorophyll molecule, reducing it to its original stage. A second photochemical process uses electrons from water to reduce NADP. As the electron is carried to NADP, at least one ATP molecule is generated. Since this process uses more energy, it requires two photons, which are absorbed by two separate chlorophyll molecules. During the next phase of photosynthesis, the free energy of the ATP molecule and electrons from reduced NADP are used to synthesize glucose. In this phase, carbon dioxide is reduced to form a 3-carbon sugar. This sugar may then be converted to glucose and other organic compounds that can be stored in the cell and utilized later to release energy. The breakdown of glucose to release energy is known as glycolysis. This process is controlled by a series of enzymes which use the free energy of glucose to generate ATP molecules. Glucose is prepared for glycolysis by accepting phosphate groups from two ATP molecules. The glucose molecule is first split into two 3-carbon sugars, which are then oxidized by NAD. In this reaction, a phosphate group is attached by a very weak bond, high in potential energy. The next enzyme utilizes this weakly bound phosphate to synthesize an ATP molecule. The 3-carbon molecule is then converted to a second activated form containing a weakly bound phosphate, which is used to synthesize a second ATP molecule. Each molecule of glucose generates 4 ATP molecules. It took 2 to start the process, so there is a net gain of 2 ATP molecules. Glycolysis requires no oxygen. Some primitive organisms use glycolysis alone as a means of releasing energy. However, oxidation by oxygen releases much more energy than is available from glycolysis alone. More evolved organisms have a system to completely oxidize the products of glycolysis. This system is known as respiration. The end products of glycolysis are carried to an enzymatic cycle known as the Krebs cycle. During this cycle, electrons carrying potential energy are transferred to NAD molecules and carbon dioxide is released. The reduced NAD molecules then transfer their electrons to a shuttle surface of electron-carrying proteins, where ATP is generated. By the end of this process, the oxidation of one molecule of glucose has led to the generation of 36 molecules of ATP, ready to do the work of maintaining life. Almost all of the energy-requiring processes of the cell use ATP as a source of energy. For example, muscle fibers use ATP directly to power contraction. ATP is also used to transport materials across membranes. Energy from ATP is also used, both directly and indirectly, to synthesize the large, highly ordered molecules that are necessary for life. To summarize, ATP is the common currency of energy transfer. The energy to generate ATP is obtained by a photochemical process involving chlorophyll, by glycolysis, the breakdown of glucose, and by oxidation-reduction reactions in the respiratory cycle. The ATP molecules are then used as an energy source for synthesizing molecules, for transporting materials across membranes, and for mechanical work. In a world in which disorder constantly tends to increase, life is maintained as a highly ordered system, low in entropy. This is possible because the ATP molecule transfers solar energy, which otherwise would be randomly dissipated, and makes it available to maintain life. And to power its activities.